All right, so today we're going to go over the Neon Transfection System. So with any other technique, the first thing is to get the protocol. So this is the, we're going to be using the Invitrogen Neon Transfection System. And so note that we have different kits. So this one is for 100 microliter kits, 50 reactions. And then this is the same thing. So this is for the larger 100 microliters. This is more reactions, so just different size, same thing. And then we also make use of the 10 microliters. So this is for smaller reaction, less number of cells. So depending on what you're doing, make sure you read your protocol so you can set up your experiment. So the first thing that I did was get the instruction and protocols from the company. So if you can zoom in, it basically goes through all the information, materials needed, and also if you don't have a standardized siRNA concentration, what the company recommends. As I'm going to show you, I started with 10 nanomolar and that works perfect. But this is something that depending on the cell type that you're using or your experiment, you have to read. And note that this is a guidebook that you can get from the website. And here I just basically, after reading all of it, these are like the pages that I mainly use. So starting at page 25. And then I'll show you page 26 basically shows you how to set it up. I'll show you eventually when I'm doing the transfections how to set up. And note that it gives you instructions of which buffers to use depending on the tip that you're using. So once again, it brings two tips, the 10 microliters, which is the smaller ones, or 100 microliters. So depending on what you're using, make sure that you use the proper buffer E or buffer E2. So that's page 26. Page 27 gets more into details of what range they give you of cells. I'll show my notes of what I use for mouse retinal microendothelial cells, but then that really depends on the person. But here's the range that they tell you of cells based on the tip that you're using. And so the smaller tip uses a smaller number of cells, the bigger tips for bigger, and that really does depend on the plate that you're using. So once again, here are the instructions of preparing adherent cells. So make sure you have this and you read it. One thing that it's important to note is that on step 10, it tells you to basically use culture media that doesn't contain any antibiotics because it can harm the cells. So once again, just download it and have this ready with you. Page 29 I like because here it's getting into the actual electroporation protocol. Once again, it tells you again to use media that has serum but no antibiotics. And now here it gives you this nice template based on what you're using, if it's a 96 well plate, 48, 24, 12 or 6, etc. Cell type, basically how much of DNA, if that's what you're using. In our case, we're using siRNA, and the neon tip that they suggest. So note that I highlight here 12 well plates because that's what I use, and for the 12 well plates I use the 10 microliter tip, and for a 6 well plate, which I eventually use to do a Western blot, I use the 100 microliter tip. And see that they suggest you can use both, but because it's a bigger surface area, I use the bigger tip. I use more cells, and I'll show in a second the amount of cells that I use. And once again, the company gives you also a range, so that really depends on troubleshooting. And then on page 30, it just basically tells you how to set up the tip. And I'll show that once I'm actually doing the transfection so you can see. And you have to make sure that it clicks, because if it doesn't, you're not going to aspirate any of your cells with your siRNA mixture. And so page 31 just continues advising that if you have bubbles on the tips, so the neon tips come in a box like this, and these are sterile, so make sure that you don't open them outside, that you open them inside your cell culture hood, and you can see what they look like. And so when you aspirate, that's actually a clear tube. If you have bubbles, then you're not going to get good electroporation. So that's what this is warning you. So definitely download the company's manual. So after I actually did my troubleshooting, these are actually my instructions for my particular cells. So if you get closer to here for the qPCR studies, I use the 12-well plates, and note that I use 5 × 10⁵ cells per reaction, and so each reaction has a total volume of 10 microliters because I'm using the smaller kit, the 10 microliter, and I put 2 transfections in each well of the 12-well plate. And I used to suspend the buffer that I used to resuspend my cells is the R buffer, and that all comes in the kit. And for the needle, I use the E buffer. For my Western blot studies, I use the 6-well plate because it's a larger plate. So for this one, I use 1 transfection, but note that I suggest using 2 so you have more cells, more material to start to do your assay. But due to limitation of cells, because you use a lot of cells, I only did 1. And so for this, I use 1,000,000 cells per reaction, and those cells were resuspended in buffer R using 100 microliters of that buffer, because for this assay, the 6-well plate, we're using the 100 microliter neon tip. And then for our buffer E, I use the E2 buffer. And so more details on the systems that I use. So we have here cells that I use are the MRMEC, or the mouse retinal microendothelial cells. I use them at passage 6. These are primary cells. And for the 10 microliter neon tip, I use 5 × 10⁵ cells per transfection. So here I have it right there. The plate that I used for this was the 12-well plate, 2 transfections reaction per well in the 12-well plate. So these are the ones that I use for qPCR. The buffer that I use, so our buffer to resuspend my cell pellet. And then for the neon tube buffer, I use 3 mils of buffer E, and once again that's for the 10 microliter neon tips. And for my siRNA, I use 10 nanomolars of the siRNA for VCAM1, and this is specific for the VCAM1 mice. And anyways, this is more details for what we're using. This you have to basically update based on whose company's siRNAs you're using. And then for the actual software, the Neon software, the machine, which I'll show you in a second, the parameters that I use were 1400 volt, 20 ms, and 1 pulse, and that was it. Okay. So the siRNA that I use is the mouse VCAM. This is from QIAGEN, so this is the information that we use, and it tells you basically how to resuspend them and at what concentration that is, and you can do your calculations. But this is what I'm using for gene silencing the mouse VCAM1 gene. Okay. So this is the machine. It's inside the cell culture hood. The on buttons in the back. So when you turn on the machine, the buttons on the on buttons on the back, it does this. And so this is connected to this other part of the apparatus. So this is the one that holds the tubes, and so the kit brings these tubes that goes in here. So see how there's that like golden little part? It goes like this, and it makes a little clicking sound. So here's where you're going to add your 3 milliliters of your buffer. So what the kit brings are your neon tips. So right here we have our 10 microliter tips that you can see. So once again, I'm opening them inside the cell culture hood. Do not open them outside. the buffer E, because this is for the 10 microliter tips. If you're using the 100, you have to use buffer E2. And then buffer R is what we use to resuspend our cells. So this is actually the needle. And so the way how this goes is that you want to click down. See how you see that little part come out? So when you're getting your tips, right, you press down and get closer so you can see it. And you are going to get your needle or your tip, and there we go. So let's do this again. So if you don't do that, what's going to happen is that part on the tip, it's not going to see how there's that golden part. If you have something like this is telling you that it's wrong. So what you need to do is make sure that when you're getting your tip, that you first press down, put it on the needle, and then release, and see how now it's clear compared to, let's say, this one. Do you guys see the difference? I don't know if you can see it with the background of my gloves.
Yeah, I can see.
Okay, so if it's like this, it's wrong. You can see the golden part. You want to make sure the golden part's right there. So make sure you do that correctly when you are going to get ready to do your transfection. So see, the golden part is down there. Last time, you press down where you see the little part come out. Make sure it clicks and then release, and then that golden part is pulled up into the needle. Okay? And then once you have your, let's say, your needle here, let's do this. Then you have your buffer. You aspirate your cell suspension with the siRNA. And so the orientation is, see where this part back here is? It goes oriented this way. And it's gonna make a clicking sound as well. Once that's ready, you set up your software. So this is already set up to what I'm using, 1400 volts, 20 millisecond, 1 pulse. You just press start. I don't do it right now because this is just a demonstration. So if I press now, it's gonna say error because it's not reading anything. It needs that buffer E and obviously your cells. And once you're done, you can just throw it in the trash. You can use it 2 times. The needle, and that's also in the protocol, so download the protocol. So once again, you can see here the needle when you press down, release. See how it's down there? When you get the needle, it pulls that up. So, okay? So make sure you do that correctly. Okay, we're gonna put it here for later use. And that's it. Okay, so here I'm gonna go over the calculations that we did so we know how many reactions we're going to need. So we're using the 12 well plate, and per each well we're doing 2 reactions. So we have our control, so 2 reactions in 1 well, and then 2 transfection reactions for the experimental gene silencing, in which we use the siRNA VCAM. Note that our stock concentration is 10 micromolar or 10,000 nanomolar. So then what we decided to do is that even though we're going to be using 2 reactions, we're going to prepare and calculate for 3, and that is because it's good to always have an extra reaction to make sure that we don't run out of volume. And that happens when you're mixing, transferring things. There's always a little bit of loss. So if you add an extra reaction or 2, depending on how many you're doing, that protects you, gives you that protecting buffer so you don't run out of solution. So for every reaction or transfection reaction, we're using 5 times 10 to the 5 cells per reaction, and we're multiplying 3. So this is per condition. So we're going to prepare 3 reactions for our control. So that's a total of 15 times 10 to the 5 or 1.5 million cells. And then we're going to use the same amount of cells for our experimental siRNA VCAM. Okay, so the next question is how much siRNA VCAM1, in our case we're using this, so you, depending on what you're using, that's, or you're trying to silence, that's what you're going to be using. So do I need per reaction? And so in each well, we have a milliliter of our media that has no antibiotics. So 1 milliliter is 1,000 microliters. And we multiply that with 10 nanomolar because this is the final siRNA concentration that we need for our experimental transfection. And we divide this by the concentration of our stock siRNAs, which is 10,000 nanomolar. So that gives us a total of 1 microliter. So from our stock siRNA, we're going to use 1 microliter to do our transfection. Okay. The next question is, remember we have our cells that we detach and then we spin them down. And so we resuspend our cell pellet in our buffer. So how much our buffer do I need to use if I'm calculating for 3 reactions? So we know that we use 10 microliters of our buffer per reaction, and we have 3 reactions per experimental condition. Therefore, 10 microliters times 3 reactions gives you a total of 30 microliters of our buffer that you're going to use to resuspend your cell pellet. So today we're going to continue with the transfection of the siRNA VCAM. So I have my protocol ready. The next thing that I do are my calculations. So because I already did all the work of resuspending my siRNA, I know that the stock concentration is 10 micromolar. That's what I have it at. And I'm using the 12-volt plates. I use 1 milliliter of media. And we're adding 10 nanomolar of our siRNA. So 10 micromolars is the same thing as 10,000 nanomolars. So we do our calculations. 1 milliliter of our total media in the 12-volt plate times 10 nanomolar. That's the final siRNA concentration that I'm going to use per transfection, divided by the concentration of my stock siRNA. So I'm going to be adding 1 microliter of my stock siRNA to each reaction. And so the cells that I'm going to be using for transfection, since I'm using the 12-volt plate, okay, I'm using 5 times 10 to the 5 cells per reaction, and I'm going to do a total of 2 reactions. This is going to change depending on how many reactions you're doing. So for this experiment, I'm only doing 2, so that gives me 10 times 10 to the 5 cells, or 1 times 10 to the 6 cells. So that's how many cells I need in order to do 2 transfections of my siRNA VCAM. Alright, so the first thing that I'm going to do is add my attachment factor to my plate. So for these guys, I only use 300 microliters of attachment factor. You don't need that much, you just need it to cover the bottom. So that's 300 microliters. If you look closely, you just mix it well that it covers the bottom. You can use 500, it's fine. You just need it to cover the bottom of the wells fairly well, so you can see right there. And that's it. I'll let that sit for 10 minutes. And note that it's important when you're going to do an experiment that your plates are labeled. So I have the 2 wells that are going to be my control. The ones that are going to have the siRNA for gene silencing. I have my cell type, passage, date, and obviously your initials. So, you know, if there's multiple people using the cell culture, you guys don't confuse your plates. So we're doing 2 of these. I'll go ahead and add the attachment factor to that as well. And things that I like doing, I like preparing ahead of time, meaning that I make sure that all the things that I'm going to use are in the cell culture for easy access, so I'm not in the middle of my experiment looking for my reagents. So what I've already done, and I'll show you, is I already have my tube, my empty tube for my cells that I'm going to be splitting. I also have my tube ready to count my cells, and I have my siRNA ready. So I have it right here. I know what concentration it is and it's ready to go. And I also have the Invitrogen Neon Transfection System ready to go. So that's that. So I have my cells here ready. I'm going to wash them and split them. Put it down. And you wash the cells twice with your PBS. Make sure your PBS is not cold so it doesn't stress your cells. Okay, now I'm gonna get my triple LE.
Oh, sorry.
Second wash. Make sure you don't actually touch the cells. Everything's done at the corner. Now we can get rid of this so I can have space. And I just need 1 milliliter of my triple LE. You can add 2 if you want to. We have plenty of cells, so I'll probably just add 2. You just need enough to cover the surface of your plate. And then it goes in the incubator for 2 minutes, no more than 5 minutes, because the triple LE will kill your cells. So we're gonna go ahead. Okay, so it's been 2 minutes. We're going to check under the microscope. So because these have a good confluency, they're still stuck, so we might give them a little bit of a tap in order to make sure that they detach. Once again, we don't want to leave them with the triple LE longer than 5 minutes. I mean, you could, but you risk them getting stressed and dying. So we'll probably put them another 2 minutes because they're still pretty attached to the plate. So we're going to put them in the incubator and then check within 2 minutes how they're doing. Wow. So it's been another extra 2 minutes, so you can see now that they're definitely more detached. Doesn't hurt to tap it a little bit more because we want to get as many cells as possible since this experiment uses a lot of cells. But you can see how they are now detached. Even though there's still a few that are attached, we're going to use the media to try to detach them, and we're ready to go ahead and collect our cells. Analyze the triple LE with our complete media. And I already have my tube where I'm going to put them. So once again, you use the media to detach them from the plate. So move it around. You want to have a good homogeneous cell culture mixture. And you can do this 4 times each time in each side, however you want to do it. You just want to make sure you detach as many cells as possible. Okay. And I always like adding them into the tube in a circular motion to make sure that they're well resuspended in the media. Now, if you're doing this for your first time, you want to make sure how well you detach your cells. After you're done, since this is trash, you can check it under the microscope. So let's check and see how much of those cells detached. So you can see that there's very little on there, so pretty good. So now we're going to count our cells. I already have my pipette at 50 microliters ready, so we're going to add 50 microliters of our cell suspension and 50 microliters of our trypan blue and count them. And so one thing to do is that you want to make sure that they're well suspended, your cells in the cell culture media. So, you know, a couple times, 3, 4, 5 times, whatever your heart desires. Make sure you mix them gently. Don't overdo it because you will kill your cells. So I'm satisfied with that. I'm gonna put this guy here on the side and take my pipette and get 50 microliters. Okay. Make sure there's no bubbles. I have a little bubble. Let's get rid of that bubble. Okay, there we go, beautiful. Put it all the way at the bottom of your microcentrifuge. We're gonna get a new tip to do our trypan blue, load them and let the machine count, or if you're using the hemocytometer to count, whatever your lab uses. You've seen the dual chamber cells, these guys from Biorad, and note that it has a and a B, so you can do 2 different cell samples. I like counting my same sample twice and taking the average. I have my trypan blue that comes in these little packages, but you can buy trypan blue, you know, pretty much order it from anywhere. 50 microliters, that's what I need. My cell counter is ready, so see where it says cell count, so it's ready. And now we add our trypan blue and mix it gently. 1, 2, 3. I think this is enough. And now you load it on onto the slide. So that's once, and you don't need a lot. You probably need like about 10 to 15 microliters. Okay, put this aside. Now we're ready and we count. Okay, so in order to determine how many milliliters of my cells that I resuspended I need, I did a cell count. I did 2 of them of my same sample. I take the average, so you add them and divide them by 2. That's my average. And so the total amount of cells that I need are 10 times 10 to the 5, which I have up here. Total amount of cells, no milliliter, because that's just the total amount of cells that I want. So how many milliliters of my resuspended cells I need? I basically divide 10 times 10 to the 5 cells by the concentration, the average concentration, 1.63 times 10 to the 5 cells per mil, and it's telling me that I need 6.134 milliliters of that cell suspension in order to have this amount. I always like rounding up a little bit more because you do have some cellular death, so I'll probably just round to 6.5 mils and call it a day, and then I'll continue my transfection. Okay, So now that I've done my calculations, I know how much of my resuspended cells I need. I'm gonna go ahead, once again, gently make sure that you mix them well. Up and down a couple of times. I like also moving the pipette tip, or in this case the serological tip, in a circular motion 2 more times, and then I collect my 6.5 milliliters because I'm rounding up a little bit. Perfect. Okay, put this here. So we're gonna spin for 5 minutes. So we're using this centrifuge. The unbutton is right here on the left. And so I have my balance. Normally I write balance so I know that this is my balance and don't confuse them with my actual cells. So B for balance. So let's go ahead and start this. Stop lid. So you put stop lid so it opens. We have a balance here. So this one is 6.5 as well as what my cells have. You put them there, make sure that they're well balanced. And then all we need is 5 minutes to spin. So it already has a timer. That's it. While our cells are spinning for 5 minutes, we want to remove the attachment factor. And this guy, we can reuse it. So note that I'm removing it from the 12 volt plates and putting it back into the tube where we store them at 4 degrees. And I'll do that for both plates. Because it's been already, we let them incubate for 10 to 30 minutes at room temp. So these have been incubating for a little bit over 10 minutes. And then to this, we're going to add our media containing no antibiotics while our cells are centrifuging for 10 minutes. So I'll go ahead and do that. So I have here my media with no antibiotics. And I only need 1 milliliter per well. So 1, 2, 3, 4. And then what I like doing while I'm working with my cells, because they have to be resuspended in our buffer and then add the appropriate volume of the siRNA, is that I put these back in the cell culture incubator so the media stays warm. So we want to keep these cells as happy as possible because this process is pretty aggressive to them. So this is after we centrifuge our cells. Can you see the pellet down there? It's that little white strip that you see. Okay. Make sure that you clean it before it goes inside the cell culture hood. And then very gently, we're actually going to aspirate the media without disturbing the cell pellet. And then we're going to resuspend with the resuspension buffer. It's 10 microliters per reaction. I put a little bit more, 22, just to make sure that when I'm resuspending the cells and there's bubbles, that I have some extra volume. Wrong tube. And so very carefully remove the media without touching that pellet. I like tilting it so I can stay away from the pellet. I don't know if you guys can see it. Okay. And then what I'm going to do is that manually I'll take that last little bit so I don't aspirate my pellet. Okay, once again, tilt it. Make sure that you don't touch that pellet. We're gonna leave it at that. And now I'm going to go ahead and resuspend my cells in 22 microliters. I'm adding like 2 microliters extra of volume. Okay, that's 22. And now you resuspend them gently. What I like doing is I like transferring them into a smaller microcentrifuge tube just so I can see my cells. So I'll go ahead and write cells up here so I know they're here, ready to go. I'll put them here. And you want to make sure that you do this gently as well. You don't want to be too aggressive with them. Okay. Put him here. Let's do that last bit. Put him here. Okay. So we'll leave him there. We can get rid of this stuff that is trash. Now let's start preparing the tips for the siRNA. Leave that there. And what we're going to do, so this is the needle that we're going to be using with the 10 microliter tips. The first thing that we need to do is add the buffer E to this tube right here. And since I have my instructions, I know that it's 3 milliliters in this tube. So I'll pull it out like this. Put that there. And so I align my tip where I can see 3. Take your 3 mils. Add it to your tube, and you can use this for up to 10 reactions. So make sure you do read your protocol because it's stipulated that you can use the tube for 10 reactions, and the tips you can only use them for 2. So my buffers have been used. Let's move this out of the way. My machine's already set up at voltage, so 1400 voltage, 20 milliseconds, 1 pulse. That's all you need. Once we're ready, let me move this out of the way, put this out of the way, move this out of the way, and then we'll go ahead and do the first nucleofection. I'm gonna go get the plate. I have our plate here with our complete media, cell culture media with no antibiotics, 2 for control, 2 for siRNA. So the first thing I'm going to do is just do the control one. So I'm gonna go ahead, and if you remember for this, you want to press down, and you see this little guy that comes out to grab your tip. If you don't do that, that metal part in the tip won't be pulled up, and so when you're using it to aspirate your cells, you won't be able to aspirate anything. So once again, you push down, go straight down, makes a clicking sound, release, and see how that metal golden part is on the top. So I have my cells here. You can like gently like tickle the tube. Don't be too aggressive with them. You can also use this. See how there's that like metal part that goes down? That's the one that you want to make sure when you're getting the tip that you remove out of your way. And so just gently swirl up and down. You can even go up and down a couple of times. You just want to make sure that your cells are well resuspended. Be gentle, okay? No bubbles. And then gently release the plunge. So I'm going to show that from far away. So we already mixed them. Gently release the plunge. Do not let that plunge go. Okay? And now what we do is that we check that there's no bubbles. I don't see any bubbles. So this is ready to go. This side goes towards the back, like this. It makes a clicking sound. Once again, make sure that the tube with your 3 milliliters, it has this little like metal part. This goes facing to the back. It makes that clicking noise. And your needle has this part that goes facing away from you towards the back, and it makes that clicking noise. Okay? Once that's ready, very easy peasy, you have your machine to go. You press start. And when it's done, it says complete. If there's a bubble or there's something missing or you didn't put something together, it's gonna tell you there's an error. So just press exit out. We don't want these guys being there for too long. And I'm gonna use the first well and gently, you know, in a swirling motion, release your cells. I like cleaning that needle with a little bit of media and gently swirl it. And then you're done. So you can use the same needle to do control or do another one, but I'm actually going to move to my siRNA, so I'm gonna have to change needles. And to dispense, just press really hard and that's done. Now, what happens if I don't have a needle or I'm doing something wrong? When you put your needle and you say start and the machine feels that something's wrong, it says tip connection error. Please check for bubbles or that the tip fits properly on the pipette. Okay, so if that happens when you actually have your tip and your cells, that means that you have to check that everything's well connected. Okay, so it's kind of nice that these machines help you out. All right, in another tube, I'm going to prepare my little aliquot of cells that are going to be transfected with the siRNA. So let's move these guys out of the way so you can zoom in. So I have my cells there. I need 10 microliters. So I'm going to go ahead and use this guy to aliquot about 11. Like I said, I always like having a little bit extra for just buffer. Okay, 11 microliters of my cells. Once again, mix them gently, up and down. Okay, we're gonna put them in our new tube. Okay, and now I'm going to add the 1 microliter of my siRNA, so I already have it ready. And that's because we made the calculations and we only need 10 nanomolar. So there is my siRNA, gently swirl it up. Oops. This is 10, not 1, so let's move it to 1. So always check your pipette so you don't make an error. I was about to do. Okie dokie. 1 microliter using the P10. So here comes my siRNA again. Mixy mixy. And see, it's a very tiny little volume. I don't know if you can focus. Very tiny. That's why I knew it wasn't 1 microliter. And then you put it in here gently. Mix well. Up and down a little bit of a couple of times. Just swirl it. Good to go. Now we're going to use our new needle to add it to our siRNA. So let's do this again. Get your needle. Press down. You can tell that that golden part has been removed. Press down gently. Swirl a little bit to get that siRNA mix with your cells. Once you're satisfied, once again, aspirate gently. Make sure there's no bubbles. And see, the volumes are so small, that's why I like adding sometimes a little bit more. You can do your calculations for 10-20% more, but always have a little bit more. I don't see any bubbles, so you can zoom in. Okay, once again, this part goes to the back. It made that clicking sound. You're ready to go. Press start. And when it's done, and if the machine was happy with the setup, it says complete. Press that X to exit out. Okay. And once again, gently, in a swirling manner, add your cells. I like cleaning that tip once, and that's it. And then you just push out to get rid of that tip. So we've used we've done 2 transfections with this tube. We can do 8 more. Okay, so one thing that I do once I'm ready is that I do this gentle up and down movement to make sure that my cells disperse fairly well, and we're going to check under the microscope. So here we have a well that doesn't have any cells, just the media, so there's nothing. Here we have the wells with our cells. So see how you can see them floating, right? So without cells, with cells, beautiful. And then our control, beautiful, okay? So always check, and then within a couple of hours you can come and check, see how they're doing. So incubator, and we're done. So right now we're checking our cells after transfection. So they've been in the incubator after transfection for 12 hours, so they look good. Since we added 1 million cells, we per well in a 12-well plate, it seems like the bottom of the plate is fully covered. So this is good to collect sample. All right, so for today, we are going to stimulate our transfected cells that we transfected yesterday with TNF. So note that we're using mouse endothelial cells, so we are going to use mouse TNF. And so because I went ahead and ordered this TNF and diluted it, I know that I started with the bottle brought 10 micrograms of TNF. I diluted it in 1,000 microliters of DI water, so I have a total of 10,000 nanograms of TNF in that bottle. And if we do to determine the concentration, we divide 10,000 nanograms over 1,000 microliters. So I know that the concentration of my TNF is 10 nanograms per microliter, or is 10,000 nanograms per milliliter. So I know that's my concentration. And for my experiment, we have 1 milliliter or 1,000 microliters of media in each well. We want a final concentration of our TNF in our experiment to be 10 nanograms per milliliter. And then we divide this by our stock concentration, which is 10,000 nanograms per milliliter, okay? And that gives us 1 microliter of our stock TNF that we're going to add to each well of our experiment. And that's it. Okay, so here are our cells. It's been over 12 hours after transfection, so we are going to stimulate with TNF. Note that our cells are mouse retinal microendothelial cells, so because they're mice cells, we're using TNF, mouse TNF. And so what I'm going to do right now is remove the media from yesterday. I'm going to put new media. Once again, we're using the one without antibiotics, just to be on the safe side and not stress these cells. And so the idea is that we will see if our transfection works. aspirate the old media. And now let's add new media, and then we are going to add our TNF. Okay? So make sure that you orient your serological pipette where you can see it. See 1, 2, 3, 4, 5, 10 milliliters. So we only have 8 wells, and we just need 1 ml per well in a 12 well plate. And you can zoom in if you need to zoom in so you can see what I'm doing. Okay, so we changed the media with new media that doesn't contain antibiotics. I already did my calculation, and I know that I'm going to treat my cells with 10 nanograms per milliliter. So I already have my TNF ready, and I know that after doing the calculations as I've shown you, I only need 1 microliter of my stock TNF per well. So I'm gonna go ahead and do that. Add it right now. I have my 1 microliter. And make sure that when you're aspirating anything, you always pipette vertically, okay? So we're gonna mix it quickly, and then take 1 microliter. And once we use that, we use it all, so I won't be putting that back in the freezer. So 1 microliter, and I go ahead and put it inside the media. So these are controls. Okay, 1 microliter to our siRNA right here, BCAM, and the last one. Okay, that's it. So once you add your TNF, you can swirl them gently to mix that TNF, or I always like just going up and down and side to side. You don't want to be too aggressive. Here we just added our vehicle because these guys are our control, so there's no TNF, and we're going to compare using qPCR the results of our control versus the results with our cells that have TNF, and that's pretty much it.